Chapter 5: Cell Biology of Bacteria and Eukaryotes. Here are the chapter objectives. Keep these in mind as we go through the chapter. Let's look at our first case study. Here is looking at Sharon, who was bitten by a tick and contracted Lyme disease. This disease is caused by the microorganism Borrelia burgdorferi. This disease can be detected by looking at antibodies in the blood due to bacterial antigens. This microorganism has flagella that moves it through body fluids like a corkscrew and has complex cells known as spirochetes. The flagella are attached to these motors that allow these bacteria to twist through capillary vesicles and migrate outward and gives the bullseye shape we see in this figure. This sign of infection is known as erythema migrans. Bacterial cells have distinctive characteristics that contribute to their pathogenicity and provide targets for medications to kill them. Section 5.1: The bacterial cell, an overview. Describe the structure and function of the bacterial cell wall and explain its importance as a target for antibiotics. Just like humans have our outer skin to protect us from the environment, bacterial cells have their cell walls to do the same. As these cells reproduce and divide, they reconstruct their cell membranes. As you can see from this image, the cell wall is separate from the plasma membrane. Some bacteria have an added layer of protection known as the capsule. This allows them even greater protection from our immune system and antibiotics. Drugs like penicillin and vancomycin block the formation of the cell wall. These medications can cause for bonds and protein links not to occur, which will cause the cell to die. Other types of medications like tetracyclines, aminoglycosides, and macrolides target bacterial ribosomes. Remember that ribosomes are the site for protein synthesis. By targeting bacterial ribosomes, this can cause for the cell not to produce important enzymes, receptors, or channels needed for their survival. In a previous chapter, we looked at the two main classes of cells: the prokarya, prokaryotes, and the eukarya, or eukaryotes. Bacteria and archaea fall under prokarya, and then you have the kingdoms of animals, fungi, plants, and protists under eukarya. We are eukaryotic species. One of the main differences that separate prokaryotic cells from eukaryotic cells is the presence of a nucleus. The bacterial cell on the left lacks a nucleus seen in the animal and plant cells on the right. Let's look at some more differences between the prokarya and eukarya. Number 1, the size. Bacteria have a small size between 0.2 to 10 micrometers, while eukaryotes have a wide range from 0.2 to 1 millimeter. Number 2, DNA organization. Bacterial DNA is organized in the nucleoid throughout the cytoplasm, while DNA is held in the nucleus in a nuclear membrane in the eukaryotic cells. Number 3, genome size. Bacteria have a small genome, about 0.5 to 15 million base pairs, while eukaryotes can range from 0.5 million to 20 billion base pairs. The genome of bacteria have very few non-coding parts of DNA. Eukaryotes, on the other hand, have tons of non-coding parts. With DNA, remember you have the exons and introns, and splicing has to occur to make a functional protein. Number 4, chromosome structure. Bacterial chromosomes are usually circular, and eukaryotes have linear chromosomes, although there are some bacterial cells that have linear chromosomes. Number 5, organelles. Bacterial cells generally lack membrane-bound organelles and ones derived from endosymbiosis. Eukaryotes, on the other hand, have several membranous organelles like the endoplasmic reticulum and Golgi apparatus. Eukaryotes also have organelles derived from endosymbiosis, such as mitochondria and chloroplast. Number 6, the cell wall. Bacterial cell walls are composed of peptoglycan versus eukaryotic cells, which are composed of carbohydrates. And lastly, number 7, flagella. Bacterial cells have a rotary flagella, where eukaryotic is more whip-like. Explain the gram-negative envelope structure and the role of LPS in pathogenesis. Here lists some important features of a bacterial cell. The cell membrane, also known as the plasma membrane or cytoplasmic membrane, surrounds the cytoplasm of the cell. The cell membrane is made out of phospholipids, proteins, and carbohydrates. It is selectively permeable and only allows certain materials to enter or exit. It also maintains concentration gradients from ions and nutrients. Then you have the cell wall. The cell wall is made up of peptidoglycan, which consists of sugar chains linked covalently by peptide bonds. Remember that peptides are amino acids, and covalently means sharing electrons to form a bond. The cell wall is superficial to the cell membrane. In lab, you perform a gram stain that differentiates bacteria based on the thickness of their peptidoglycan wall. Gram-negative bacteria, seen in the top right, has a single layer of peptidoglycan, while gram-positive bacteria, seen on the left, have multiple layers. Gram-negative bacteria, unlike gram-positive bacteria, have an outer membrane. Let's look more closely at a gram-negative cell. Here shows a model of E. coli whose structure can be applied to many bacteria. Due to microscopy and subcellular analysis, we are able to make these extremely microscopic images. Looking back at the outer membrane, between it and the cell membrane is the periplasm, which is a fluid that contains proteins that can allow for the transportation of carbohydrates. If you look at the outer membrane, it contains phospholipids, but also lipopolysaccharides. These are types of lipids that have long polysaccharide chains. Remember that polysaccharides are several linked carbohydrates or sugars. The lipopolysaccharides, LPS, along with other polysaccharides, can create a thick outer capsule surrounding the cell. The capsule forms this slippery mucus layer that inhibits phagocytosis by white blood cells because their pseudopods cannot hold on to the bacteria. This is what allows microorganisms like Mycobacterium tuberculosis to evade our immune system. The cell membrane, cell wall, and outer membrane form the cell envelope. Since bacterial cells lack a nucleus, their chromosomes are organized within the cytoplasm as a system of looped coils called the nucleoid. This system allows for faster reproduction and protein synthesis because transcription and translation can occur at the same time. In eukaryotic cells, transcription has to be complete before translation can occur. Describe the biochemical composition of a bacterial cell. Chemically, all bacteria share these common features. Water is a necessary solvent. You have inorganic ions like potassium, magnesium, and chloride ions. Potassium stores energy by creating concentration gradients around the plasma membrane. Magnesium is important for the function of certain enzymes like RNA polymerase, which wouldn't be able to extend RNA's chain during transcription without its presence. Small organic molecules are present, such as lipids and sugars, that form cell structures like the cell envelope and can be used for nutrition. Lastly, you have macromolecules. Examples of these are nucleic acids and proteins. Nucleic acids like DNA and RNA contain genetic information for the cell. You have enzymes, which are proteins that catalyze reactions. Then you have protein channels, which mediate transport of molecules and ions across the cell membrane. The amount of these chemicals varies with species, how they grow, and environmental conditions. As you can see from the chart, bacterial cells are composed of 70% water. The next most abundant chemicals are proteins at 16%, which are enzymes, receptors, marker molecules, or channels. The total proteins encoded by a genome and capable of expression in the cell is known as the proteome. Next is RNA, which has its components for protein synthesis. You have lipids at 4% forming the membrane of the cell, DNA at 1% containing genetic information, metabolites and biosynthetic precursors at 1.3%, which are chemicals important for creating energy and structures within the cell, peptidoglycan found in the cell wall at 0.8%, and lastly, inorganic compounds at 0.1%. Section 5.2, Bacterial Membranes and Transport. Describe the functions of a cell membrane. What defines the existence of a cell? That would be the cell membrane. The cell membrane surrounds the cytoplasm and mediates transport from the extracellular fluid and intracellular fluid. The cell membrane is composed of a phospholipid bilayer containing proteins and carbohydrates. The proteins can act as channels and transport molecules and ions in and out of the cell. Other membrane proteins can act as receptors and detect changes occurring in the bacteria's environment. The proteins found in the cell membrane are very important and have the following features. One is structural support. Membrane proteins can connect all the layers within the cell envelope. Other proteins can serve as the base of pili and flagella. Pili can connect cells to each other or grab materials outside of the cell. Flagella allow for the cells to move. 2 is detecting environmental signals. These proteins act as receptors which can cause the cell to change which proteins it creates in order to survive or cause for it to be more virulent. 3 is secreting virulence factors and communication signals. Some types of bacteria have membrane proteins that can help them secrete toxins created by themselves or medications that we use to kill them. Other membrane proteins can allow for communication among cells or the host they're infecting. 4 is transport across the cell membrane. This is what gives the cell its selectively permeable quality. Lastly is energy storage and transfer. Energy storage is created by forming concentration gradients. Explain how nutrients are transported and how energy is spent to drive transport. We call the plasma membrane selectively permeable, which means only certain things are allowed to pass it. The materials that can cross do this by different processes of diffusion. So what is diffusion? Remember that diffusion is the movement of molecules and ions across the plasma membrane from an area of high concentration to an area of low concentration. This means that these chemicals move down their concentration gradient. Nutrients like sugars can bind to transporter molecules called permease that facilitate their movement across the membrane. Diffusion can be active or passive. Molecules such as oxygen and carbon dioxide move passively, but most chemicals cannot. They have to move by active processes. The difference between passive and active transport is energy. The passive transport of chemicals do not require energy, while the active transport of chemicals do. Passive transport follows the rule of diffusion, where substances move from areas of high concentration to low concentrations. Once again, this means down their concentration gradient. Types of passive diffusion are simple diffusion, facilitated diffusion, and osmosis. Simple diffusion involves lipid-soluble or small molecules and ions that can diffuse across the plasma membrane without being hindered. Facilitated diffusion is where you have a membrane protein that facilitates the movement of the chemical from one side of the membrane to the next. And then you have osmosis, which deals with the transportation of water through aquaporins. Then you have active transport that uses energy like ATP to move chemicals from low concentrations to high concentrations. This means that it can move substances against their concentration gradient. This is why energy is necessary. We've heard about ATP being the energy molecule for cells, but there are other ways for cells to obtain energy like proton motive force, which we'll discuss shortly. In this video, we're going to look at active transport in action and review the concepts of symport and antiport.
Small molecules, such as sugars and amino acids, can be transported up a concentration gradient. The sugar moves via a membrane transport protein from outside of the cell where the sugar concentration is low to the inside of the cell where the sugar concentration is high. The transport of the sugar through a coupled transport protein is driven by the movement of counterions such as sodium ions or protons that are moving down their concentration gradients from a region of high to low concentration. Sodium ions and the specific sugar or amino acid simultaneously bind to the same transmembrane protein on the outside of the cell, called a symport. When the counterion is sodium, the low concentration of sodium on the inside of the cell required to transport the sugar is maintained by the sodium-potassium pump, which is powered by ATP. In a related process called countertransport, the inward movement of sodium ions is coupled with the outer movement of another substance such as calcium ions. As in cotransport, the sodium ions and the other substance bind to the same transport protein called the antiport, but in this case they bind on opposite sides of the membrane and are moved in opposite directions. The low internal sodium ion concentration is maintained by the sodium-potassium pump.
The transport proteins that use ATP to move molecules and ions across the plasma membrane are known as ABC transporters. ABC stands for ATP binding cassette. ABC transporters, called efflux transporters, help to expel toxins from the cell. These can be self-made or help the cell to expel antibiotics. An ABC transporter consists of two proteins that form a membrane channel and two cytoplasmic proteins that allow ATP to bind, and this is the ATP binding cassette. Lastly, there is a substrate binding protein which binds the substrate or solute. It then transfers the substrate to the channel protein. When certain nutrients or minerals are in low supply or inaccessible, cells can secrete molecules to find and obtain these chemicals. The example of these molecules are these siderophores. Siderophores are iron-binding molecules. Iron is an essential nutrient for most cells. Iron is usually bound to other atoms in the form of iron hydroxide. Since this compound is insoluble in water, it is hard for cells to obtain free iron. The siderophore will bind ferric iron available in the environment. Ferric refers to any compound that contains iron. The siderophore will bring the iron to a transport protein so the mineral can be transported inside of the cell. The last thing in this section we're going to discuss is proton motive force. Earlier we spoke of active transport that requires energy for materials to be transported across the membrane. We spoke of ABC transporters that uses the energy molecule ATP to transport these molecules across the membrane. ATP is not the only way cells can obtain energy for transport. Another important process is proton motive force. This deals with stored potential energy from a cell's metabolism. Chemical energy is used to pump protons, essentially hydrogen ions, outside of the cell, increasing the concentration gradient. As these positively charged ions move across the membrane, an electrical gradient is produced, making the outside of the cell very positive and the inside of the cell more negative. This difference in proton concentration and charge can drive the movement of nutrients into the cell by transport proteins, energize the motors that rotate flagella, and cause the synthesis of ATP. Some antibiotics, like diaroquinolin, target proton-powered ATP synthase, which can be seen in this image. Section 5.3, the bacterial cell wall and outer layers. Describe the cell wall structure and explain how it protects bacteria from osmotic shock. How do bacteria protect their cell membranes from falling apart under pressure? When bacteria infect their host, they are rapidly introduced to a new environment that can have a different temperature, pressure, and solute concentration than their previous environment. The cell wall helps bacteria to deal with their changing environments. The cell wall makes a bacterial cell rigid and helps it withstand intracellular turgor pressure that can build up as a result of osmotic shock. What is turgor pressure? As water enters the cell, it creates an internal pressure. Turgor pressure deals with the pressure caused by the entry of water that pushes the plasma membrane against the cell wall. High turgor pressure can be created if the cell is placed in a hypotonic solution and water enters the cell. Low turgor pressure can be created if the cell is placed in a hypertonic solution and water leaves the cell. The cell wall is very porous to ions and small organic molecules, unlike the cell membrane. The image to the left shows an entire cell wall isolated from E. coli. In the right image, you can see how the cell wall is constructed. You have glycan chains made out of carbohydrates. These form parallel rings that are cross-linked with amino acids, forming a peptide cross bridge. Most bacterial cell walls are composed of peptidoglycan, which is unique to bacteria. This is why the cell wall is a good target for treatment for a bacterial infection. Since peptidoglycan is not found in human cells, there is a low risk for side effects. Peptidoglycan can also be called murine. Drugs that target the peptidoglycan cell wall key in on the enzymes that causes its construction. By destroying these enzymes, you destroy their protective outer covering and lead to the death of the pathogen. Let's look more closely at the cross bridges and two antibiotics that interfere with its functioning. Here shows alanine, glutamic acid, diaminopimelic acid, making the peptide cross bridge. Penicillin attacks and destroys the enzyme transpeptidase. This enzyme creates the formation of the cross bridge between the glycan chains. Another antibiotic, vancomycin, blocks the formation of the cross bridge by binding to one of the amino acids, locking it up so it cannot bind. Both of these interferences lead to the destruction of the bacterial cell. Due to the overuse of antibiotics, bacteria have adapted and become resistant. Penicillin-resistant bacteria created an enzyme beta-lactamase, which breaks apart penicillin, making it non-functional. Vancomycin-resistant bacteria also created an enzyme that added a chemical to the end of the peptide branch, which blocks vancomycin from locking up the terminal amino acid.